Hello and welcome to me and AJ Ottomano's presentation on DNA replication. The major step for DNA replication to take place is the breaking of hydrogen bonds between bases of the two anti-parallel strands. The unwounding of the two strands is the starting point. The splitting happens in places of the chains which are rich in iodine and thymine. That is because there are only two bonds between iodine and thymine. There are only three hydrogen bonds between cysteine and guanine. The helicase is the enzyme that splits the two strands. The initiation point where the splitting starts is called the origin of replication. The structure that is created is known as the replication fork. One of the most important steps of DNA replication is the binding of RNA primase in the initiation point of the parent chain. RNA primase can attract RNA nucleotides which bind to the DNA nucleotides of the strand due to the hydrogen bonds between the bases. RNA nucleotides are the primers for the binding of DNA nucleotides. Step 3. The elongation process is different for the template. The preceding daughter strand that uses a 5'3" template is called a leading strand because the DNA polymerase A can read the template and continuously add nucleotides. The template 3-5 cannot be read by DNA polymerase A. The replication of this template is complicated and the new strand is called the lagging strand. In the lagging strand, the RNA primase adds more RNA primers. DNA polymerase A reads the template and lengthens the bursts. The gap between RNA primers is called Okazaki fragments. The RNA primers are necessary for a DNA polymerase A to bind nucleotides to the three end of them. The daughter strand is elongated with the binding of more nucleotides. Finally, you have step 4. In the lagging strand of the DNA pol 1 reads the fragments and removes the RNA primers. The gaps are closed with the action of DNA polymerase and DNA ligase. Each new double helix is consisted of one old and one new chain. This is what some call semi-conservative replication. In the last steps of DNA replication is the termination. This process happens when the DNA polymerase reaches to an end of the strands. We can easily understand that it is in the last section of the lagging strand when the RNA primer is removed. It is not possible for the DNA polymerase to seal the gap. So, the end of the parental strand where the last primers bind isn't replicated. These ends of linear DNA consist of non-coding DNA that contains repeat sequences and are called telomers. As a result, a part of the telomere is removed in every cycle of DNA replication. The DNA replication is not completed before a mechanism of repair fixes possible errors caused during the replication. Enzymes like nuclease remove the wrong nucleotides and the DNA polymers fill the gaps.